Hello, students. Welcome to the first week of our course. And today we're going over chapter one of your textbook. That is the study of life, which is biology. Biology and the study of life. And more specifically, we're going to dive into what is science. Now, just to give you a little idea of what these lectures will look like. Um, I do have the ability to write and point out stuff. So if specifically I want to, point out a word or circle something or give you some insight onto a particular formula, I do have that ability. So we're going to use that. I can also, if I want to, change color. I can draw arrows, so on and so forth. I can go to a white screen. I can go to a black screen. It's a lovely little feature that I have here that we're going to use. So, um, yeah, let's dive into it. So, the study of life, is what we're going over today. Biology. That's what you're all here for. But more specifically, if you take a macro view of everything, um, what is science? And I don't know how many people can accurately define this or what it actually entails. We do live in a generation that kind of science is like this catch-all philosophy. You can't escape like Instagram or any other social media without somebody saying research shows, but the bulk of people I don't know understand what science is. So what is science? That's what we're going to talk about today. What is biology? That is what we are going to talk about today. You already know a little bit about me. This is my name is Chaz. And again, it's Chas, Chaz with a S, and my last name is Stevens with a PH. So you can call me Chaz, Professor Stevens. I don't mind. But my name's Chaz Stevens. This is actually not my first semester adjunct teaching. It is my first semester adjunct teaching online, however. So we're going to learn together here. I am married with three kids. I have a 17-year-old, a 2-year-old, and a 1-year-old as of this recording. As you know, I have my master's in biology and statistics. I work at the School of Nursing. Um, I am a published author, so I have some research out there with my name on it. Um, I'm an avid sports fan. I'm a Cowboys fan. Love my Texas teams, but I watch Cowboys every Sunday. So when football season's on, so you may be checking out of this course already because you know that. Please don't. Know that I suffer for being a Cowboys fan enough. I am old enough to remember their last Super Bowl though, so take that to heart. And I also love to play chess. I don't get to play as much as I used to, but I still love to play. So I'm always up for a good game. So today we're going to be talking about biology. We're going to define biology, its importance. Um, what are the characteristics of life? So when we're looking at the characteristics of life, what does it mean to be alive? So if I say something's alive, what does that mean? Um, we're going to look at the scientific method. The scientific method is something that I think a lot of you know about, or at least have heard about. Maybe you don't know everything about it or understand everything, but you've at least heard of it. We tend to look at the scientific method or the scientific method is something that's explored in, I think, middle school, high school to some level, but the the theory and the methodologies are still the same. I want to look at the types of reasoning that we look at whenever we look at observation. What do I mean by this? Well, uh, every type of observation and conclusion drawing mechanism uses either inductive or deductive reasoning. So inductive or deductive reasoning, we're going to look at that. And you should be able to distinguish between those two. I'm also going to present to you some ethical issues that we see in science and why we do things the way we do them. And we're going to describe biological organization, the hierarchy of life here. So biological organization, that's a terrible straight line. I apologize. And then we're going to look at some of the major branches of biology. So keep that in mind as we go through this. And again, After this, if you still don't understand everything I'm talking about, feel free to shoot me a message to my email address. So I want to start off with this quote. It's a quote by Sir Peter Medawar. He was a Nobel laureate, and this is what I want to start you with. This quote reads that there is indeed a limit upon science is made very likely by the existence of questions that science cannot answer, and that is in no conceivable advance of science, and that no conceivable advance of science would empower it to answer. So notice it cannot answer, and no advance would allow it to answer. So he goes on to say, it is not to science, therefore, but to metaphysics, imaginative literature, or religion that we must turn to for answers having to do. Sorry, it is not to science, therefore, but to metaphysics, imaginative literature, or religion that we must turn for answers to questions having to do with first and last things. Now, take that quote for what it's worth, but what I want to point out is that he has this idea that there is a limit upon science. Science has limits. In other words, there's some things that science cannot do. Let me get my eye up there. So science has limits. There's a limit upon science. There are questions you can ask that science cannot approach. It cannot answer. So what does science, or where do those limits exist? I want to touch on that, but let just kind of stew on that quote for a little bit. Again, Sir Peter Medawar, Nobel laureate, made this quote many, many, many years ago, long before any of us were born, um, that science has limits. Let's proceed and let's ask this question. What is biology? So we have science, we have limits. So what is biology? Well, biology, if you break down the word, let me get a different color here just to show you how fun this is. We have it broken down into two words. Bio equals life, ology equals study. So you have biology, the study of life, science of life right here. Biology studies living organisms, interactions. And again, biology is that umbrella term that we see. Let me get my eraser out here and get rid of that because I don't like that line. It's a terrible line. Um, so let me get my ink. And so, yeah, so we come here and we just say it's the science of life, living organisms, interactions. A branch of these interactions is something called ecology, which is a branch that I was very familiar with in my graduate studies. Um, but anything that's living falls into this category. And now this can encompass everything from microscopic cells, ecosystems, again, ecology, the full spectrum of life on Earth. What's important about this is it's very relevant to what we have today, to our life today. Um, we see biology in every single day. We see them in disease outbreaks. Anybody remember COVID? Yeah, it was a terrible time. That was a viral outbreak. Medical breakthroughs. We're seeing new and new, more and more advances with cancer research, and the like. So it's very relevant to your life, but also to understand the mechanism by which we get to these breakthroughs and to these outbreaks is very important too. They all connect to biology, though. So you don't have to look to COVID or to current day or current to modern phenomena to explain these or to show relevance. You go look in the past, you had the Black Plague was another outbreak. You had the Spanish flu. You had all these different outbreaks, medical breakthroughs. You had and we'll talk about it in a little bit, like penicillin was a medical breakthrough that we are living in the ramifications and the benefits of these discoveries. So just keep that in mind as we go through that biology touches everything more than you think. So just a quick knowledge check, and you can pause and answer this question. But let's say a person comes up to you, a friend, a family member. Says this topic isn't biology because, and they quote, it doesn't involve humans. So which response best corrects them? Now you can pause and take your time to find the answer here. So go ahead and pause it. So now that you've paused it and you found your answer, the correct answer is number two, is that biology focuses on living organisms and how they interact with each other and their environments, regardless of whether humans are involved. Now, again, we talked about this. You can have bacteria, viruses, uh, other animals. I'll say other animals because humans are, in fact, animals. Um, so yeah, so that just because it isn't humans doesn't mean it's not biology. Um, biology includes organ. Let's look at answer one. Biology only includes organisms that are large enough to see without a microscope. That is not true. Bacteria and viruses thwart that plan. Biology is mainly the study of medical treatment and diseases. No, remember I said you have ecology. And biology includes only experiments done in laboratories. That's just a nope. Botanists and other people and do stuff in the field. So um, the answer being two, anything that has life. And we'll get to defining what life is here in a minute. But like I said, you have disease outbreaks, medical research, environmental action. All this connects to the day-to-day life that you experience. So, question: what do you think these are photos of? Um, look closely, take a few seconds, pause it if you want to think about it, take a few guesses. When I present this, I see a number of answers here. Bacteria, viruses, I've had snot, boogers. None of those are true. This is actually photographs of pollen. So yeah, you heard me right there, pollen. These are taken with a what's called a scanning electron microscope. Electron microscope. Um, and if you look closely here, it's hard to see, and I'll get um, the highlighter here to kind of show you. Like, look, if you look right here, and right there, you'll see that it says SEM, that's scanning electron microscope magnification, 3.56 thousand for the one on the far left, 3.68 thousand on the middle one, and 4.71 thousand. Um, magnification. So these have been magnified 4.7 thousand times um, to see what you see here. Now, the next question is, uh, sorry, let me get my better pen here. My pen, get my color here, change to blue. Now I want to ask this question. Who took these photos? And I'm not going to waste the suspense. Me. Back in 2006. These are 5 photos from a research project I did on independent research project. But the observable world that we look through, or the observable world with advances in technology, has allowed us to see many different forms of life, um, such as pollen. And you can go even further down here. If you have a transmission electron microscope, you can see bacteriophages. You can see all kinds of stuff. So just a glimpse into what we see in this world. It's pretty astounding when you think about it. So again, just what we can see is always changing. What we can observe is always changing. So the idea of life is pretty vast. Um, so moving right along. All right. So I want to ask this question to you. This is a thought game. It's prompted and it was presented by a man named Carl Sagan. He's a scientist from the 70s, back in the day, long time ago. But he postulated science with respect to an invisible dragon, and he kind of like made this parallel to what is science, what isn't science, and we're going back to that question. So when you're thinking about this, what is science. Think of the invisible dragon. Okay. So Carl Sagan has a parable of an invisible dragon, and it's in a garage, but it can be anywhere. The idea behind this is I'm going to make a claim, and I'll make a claim that should promote thought, or it should promote a question. And basically says, I have a dragon in my garage. Okay? I have a dragon in my garage. I am telling you this. Naturally, this should bring up questions. Um, One of the questions could be, okay, if you have a dragon, where is it? Right? That's a logical question. Where is it? Or can I see it? Well, unfortunately, my dragon is, as Carl Sagan says, it's invisible. You cannot see this dragon, but he exists. I'm telling you, he exists. Your next question could be, well, how can I prove, or how can you prove this exists? And you come up with ways to test this. You think that a dragon breathes fire. So, um, this is another thing. I can't see the dragon, but he breathes fire. Um, so let me just kind of write this out here so we see where we are. Fire. He has fire. So maybe you're thinking, okay, maybe I can get a thermal gun and test the temperature. Okay, well, the fire that this dragon has doesn't have any heat. Okay, so your test is going to be moot. You're not going to be able to see the fire because it has no heat. Okay, so I have an invisible, heatless dragon. Well, what about, well, can I touch him? Um, to which my response would be, oh, he's incorporeal. Let me rewrite that. Incorporeal, which means he has no body. So you can try to go touch him, but your hand's going to go right through him. Okay? So now I have an invisible, heatless, incorporeal dragon. Um, the point of this whole exercise is that with every single question I have to validate your claim, um, I am unable to falsify or test your claim because there is no test. So one of the traits of science, of good science, is something must be observable. It must be testable, and it must be repeatable. Okay? So I have to observe it. So with your dragon claim, or my dragon claim, I should say, what you weren't able to, you can't observe it. You can't, you proposed a bunch of tests, but with every single test, I struck down. You can't touch him. You can't see him. You can't measure his heat. So this does not fall into the realm of science. Now, this is the limit that Sir Peter Medawar was saying is like, just because we can't see it, we can't observe, we can't test it, it's not science. So there are certain things that we think about in our day-to-day life that aren't testable, that don't meet this criteria. One of the biggest things is anything pertaining to history. History falls into this claim. So we cannot test the Gettysburg Address. It is not observable, testable, or repeatable. We have to rely on a different account. That is historical stuff. What we're relying on here is the observable, the testable, the repeatable. This is the idea of Carl Sagan's invisible dragon and how it really does quantify and tie in science pretty nicely. Now, I will say this. I want you to remember Carl Sagan's invisible dragon because it will show up on your midterm. This is something that's big. I am big on this. Okay? So remember that. Okay? Let's clear our notes here and move right along. So the scientific method. When we look at the scientific method, we are looking at a series of steps to understand the observable universe or observable world. It usually starts with an observation. More often than not, it starts with an observation, which then proceeds to a question, which then proceeds to a hypothesis, which then proceeds to an experiment, and finally analysis. And analysis is where statistics come in, if you were not familiar with that already. And this is where Chaz comes in a lot of stuff. So, in case you were wondering what I do, there is a branch within research called experimental design. I help people with this too. So, designing your experiment so that you can show your results as best as possible is something that I do. So again, observation, question, hypothesis, experiment. These are the steps. You have to observe something. Why did that happen? And then you have to propose a testable explanation for the observation. Usually it has to be quantifiable in some way. So it has to have some sort of number associated with it. It could be like a test score, could be weight, whatever. It needs some form of testing that you can do for that. Then with your experiment, you design, you get and you get your results from that, and you analyze it. And then hopefully you do a good enough experiment, you get it published. So moving right along here. I'm never going to ask you to memorize people's names because I really don't. I think it's good barroom trivia, but at the end of the day, it is in your book. So the guy who came up with this, his name's Sir Francis Bacon. I don't care if you know him. I won't ask about him, but it's in your book. So if you want to commit it to memory and try to win a barroom trivia night, go for it. So I'm going to throw you out a little knowledge check here. Okay. Designing an experiment. A researcher notices a sudden drop in fish population in a lake. They hypothesize, there's that word, that agricultural runoff is increasing algae growth, which then reduces oxygen level crucial for fish survival. What experiment could the researcher design to test his hypothesis, and what specific controls would be essential for valid results? Explain your reasoning. So we really haven't talked about this yet, but essentially, our scientist here, or we'll say you notice that there's a sudden drop in fish population. So at one point we had 100 fish or something, and then all of a sudden now we have 25 fish. It's a drop off. Why is this happening? There's our observation, OBS, observation right there. 100 fish to 25 fish. So a researcher notices a sudden drop in fish population. So they think, misspoke there. They think, hypothesize that the agricultural runoff, i.e., so this image up here, you see, let me change my color here so you can kind of see. Um, let's go to, we'll go to yellow up here. They think that this stuff up here is running into the lake here and causing some fish to die. There's your dead fish right here. Okay. So this stuff up here from this agricultural runoff could be pesticides, whatever, is causing this to run off and kill the fish. So what experiment could you design? Well, one thing you got to do is you got to have a control. And we really haven't talked about this, but in any experiment, you have to have a control, which is another way of saying this is something that has no change. It's a comparison tool. And you also have a variable group. So this is the thing that changes. And so we're talking about these two things in tandem. So in this particular case, the variable that I see, the thing I'm testing, so we can say that another way, the thing I'm testing is this runoff up here. I'm testing to see whether this runoff has an effect. How do I do that? Well, I need to compare it to something. Well, what if I were to compare it to a lake, this lake over here, where there's no runoff? If there's no runoff, and hence no algae, I should see more fish. So this is a very easy experiment to design because you could test how much runoff you have here. You have no runoff here, and you could see how that's affecting the fish population. So when I'm looking at control, I always have to have a control, something that I'm comparing to that has no change. My variable is the thing I'm testing. I'm testing this environment here that has this runoff. You need to be able to identify in any experiment I give you, what is the hypothesis? What's the observation? What's the results? What is the analysis? Is this a valid test? So this is very important that you're able to identify this. So a knowledge check. Here we go. Another one. A person claims that a new herbal supplement, and I want you to read this carefully, works only if you truly believe in it. And it cannot be tested scientifically. So why does this claim fall outside the scope of inquiry, of scientific inquiry? Well, remember the claims that you have to make. Remember the criteria. It has to be observable, has to be testable, and it has to be repeatable. Observable, testable, and repeatable. Um, how do you, here's one thing is like, in the grand scheme of science and things that don't fall under science, there are things that are observable but not repeatable, right? So like you can observe, let's just say you can observe we'll just call it, well, you can observe an asteroid hitting a planet. So we'll delineate that. You can observe an asteroid, but you can never observe that asteroid hitting that planet a second time. It is not repeatable. It's not repeatable. So that kind of falls out of the scope of science that we're looking at it. You cannot repeat that, and therefore it's not testable. So in this particular claim, why does it fall out of the scope of science? Okay, I could observe a person taking it, but can I observe belief? Can I observe belief? Can I test belief? Can I repeat it? Now, you might answer, yeah, you can repeat it, but how do you test it? How do you observe it? You don't know. So these things fall out of the scope of science. Now, this is more philosophical. It doesn't mean it doesn't work, but as far as science goes, it doesn't fall under that scope. So keep that in mind when you're thinking about what is science, what isn't. Remember, if somebody says, "Hey, research shows," you should be asking, "Well, could they observe a phenomenon? Could they test it? Can they repeat it?" That is the backbone of the scientific community of published research. Observable, testable, repeatable. There have been a number of research articles posted where somebody tried to repeat it in another lab across the country, across the globe, couldn't do it. Turns out the scientists falsified their claims. Keep that in mind. So, to kind of wrap that up, to kind of circle back to that, you have to have a hypothesis, an answer to the claim that the observation that you're making. It must be falsifiable. A hypothesis must be falsifiable. Experimental results can disprove it. Any claim needs to have the ability to be disproved through observation, measurement, and when we're looking at a hypothesis and an experiment, you're looking at two groups, a variable and a control group. The variable is the thing you're changing. We'll start with the control group. The control group right here has no change. And the variable group, we're introducing some change. So what's a good example of this? Okay, so let me get some space here. What's a good example of this? Let's just do like we'll just do, for instance, we'll do a cancer drug. That's bad. A cancer drug. Okay. I could have a control group and I could have a variable group. The control group gets no drug. The variable group gets the drug. Now, of course, both members of this group have cancer, the same cancer probably, but in this control group, they don't get the drug. In this variable group, they get the drug. Now, to make it a little bit more fair to eliminate some other variables, um, we might even do some things like say we'll give them a placebo, something called a double-blind study. They don't know what they're getting, and neither does the variable group. But they're both getting a pill, and then the even the person giving them the drug doesn't know. Only the testers behind it. It's called a double-blind study. But this is a way you can test results. If the cancer goes down in the variable group or they stay the same, you have information that can help you further your science. If they stay the same, the drug doesn't affect cancer at all. If it changes in the variable group, well, there's something to this drug. Still has to go under a lot more tests, but that's the basic gist of it. So I posted a video on D2L for you to watch. And it basically goes over the entire Galileo trial. Now, you may have heard of this story, is that Galileo makes a claim. And up until this time, up until Galileo came along, the claims that the scientific community had a good idea, what they thought was a good idea, that the solar system was geocentric. And you go watch it if you haven't. I'm going to try to sum it up, which meant that the Earth was the center of the solar system. Galileo came back and he said, you know what? Based on what I'm seeing, I think we are heliocentric, which means I think the sun is the center of our solar system. Looking back now, we know he was right. Now, the myth goes that Galileo presented his findings to the church. The church sent him to jail for heresy, and they forced him to recant. If you want to go with the Spark Notes version, um, that's fine, and it's not even all that accurate. Really, why I present this to you, and go watch the video. It's more in depth than what I have time to present right now. Um, the gist of it is that Galileo, being the inventor he was, really uh, put forth a brand new, I guess, a modification, and I would say it's just a brand new invention altogether. He had a better invention of the telescope than anybody else. So he could see things that nobody else could see. Unfortunately, when you present your science, he could observe it, he could test it, but that technology wasn't available to everybody. So, in the grand scheme of things, yes, he could observe it, yes, he could test it, but while the technology wasn't around to everybody else, especially the church, it got met with a little resistance. Now, that being said, we look back and we say, okay, he was right, understandably so. However, I just want you to kind of put it into a 21st century eyes. Supposing someone came to you and said, hey, um, turns out we don't breathe oxygen. Now, this is something we've all accepted for many, many years, and this guy comes along and says, hey, we don't breathe oxygen. Um, you would be like the invisible dragon prompt I showed you earlier. Well, can you show me? Well, only I have the tool that can see it. Interesting. That falls out of the range of science, and it's going to be met with resistance in the scientific community. Until other people can observe it, it's never going to get past that front stage. Now, there's a lot of politics that go into this, sure, but the idea behind it is that only Galileo could see what he was proposing, and no one else could. Were there other politics involved? Sure. But the myth that it was just met with such resistance because people didn't want to change their beliefs, not quite right. So he was right, but he didn't follow scientific protocol, which, again, not really invented at the time, but it's understandable why that would be met with resistance. So there's more to it. Watch the video. It's actually pretty interesting. Moving right along. Some pioneers of science. Again, this is the guy. Again, I don't expect you to remember this guy, but this is the guy that basically, and I'm not even trying to butcher his name, but he's Hungarian. Um, so we'll call him Iggy. But basically, Iggy was the guy who invented hand washing. It's hard to believe that there was a time we could go to hospitals and no one would wash their hands. Um, so this will actually shock you because there was a time back in like the mid 19th century, so you're talking like the 1800s. He was noticing a bunch of there was high mortality rates. So mortality rates right there from something called puerperal fever in maternity wards. Basically, newborns were being born. Um, so you had moms delivering babies. The moms were dying, and that the babies were dying too. Long story short, back in the 18th century, what you were seeing was you were seeing doctors going and produce and conducting autopsies. And here you go, you have the word autopsies right there. Conducting autopsies on dead individuals, and then immediately just walking over and delivering babies. They weren't washing their hands. So he postulated, like, okay, could I dip my hands? What if I were to clean my hands, dip in some chlorinated lime solution, and then go deliver the babies? And then he saw the reduction in deaths. Um, it's funny because uh, up until this time, no one thought that that would be a problem. And then he comes and says, no, you need to wash your hands. And now, we wash our hands just kind of as best practice. But there was a time we didn't do that. Um, so moving right along. So, I will ask this question in some way, shape, or form. Maybe it's going to be on your quiz. I don't know yet. But um, when we're looking at the scientific method, um, I have this catch-all term for a hypothesis. So basically, this is how the scientific community works here. So here's a hypothesis. And I'm going to go to my whiteboard here. So you have two ways of conducting an experiment. Well, two questions you need to answer. Again, observation, you go through analysis, you go through a hypothesis. Um, when you get to the hypothesis section, so let's go back to our good old cancer drug. Cancer drug. That's a, there you go, cancer drug. Okay. You have a cancer drug. And you want to ask yourself, does this drug work? Okay. So the cancer drug, does this drug work? Okay. So we're going to propose a test. And we're going to test this drug. So now in any scientific research, you have something called a null hypothesis. And a null hypothesis just basically says there is no change. So in this case, we're saying the drug does not work. And then you have something called the alternative hypothesis, which is saying the drug works. I'm going to bold that so we can see. Long story short, in the end, this is where statistics comes in. Does this drug work? I'm going to assume that, and let me change colors here so you can kind of see this better. Let's go to the blue. Does this drug work? I'm going to assume that my null hypothesis is true until I have sufficient evidence to accept this guy. So that's done with a lot of statistics, but how research is working, cancer drug. Does this cancer drug work? Until I've done enough testing, enough analysis, I'm going to go in with the mindset that the drug does not work. And I'm not going to present it to the population, to the populace, that it does work until I am very, very certain that the drug does work. Because the last thing you want is you do not want to give out a drug that you say works when really it doesn't. So another term that we use when we talk about this stuff is a false positive. False positive is one way to look at it. When I say something works and it doesn't, we'd much rather have a false negative in our we're very conservative in the scientific community. False conservative. Sorry, false negative. And we're very conservative. I was trying to write that. So the idea being is until I get enough information, I'm not going to say that there's a change in this status until there's enough evidence. So until I have enough evidence, I'm going to say the drug does not work. And once I get enough evidence, then I can say there's evidence to support that the drug works. So going back to your hypotheses here, you always have this null versus this alternative. And the null just basically says there's no significant effect. The alternative is saying that there is a significant effect. So take this into any test. Take it to that runoff test. Take it to the or that runoff example. Until we have enough evidence, the runoff isn't causing it. So we don't want to implement sanctions on that farm that's causing that runoff until we can say, yeah, you are causing it. See what I'm saying? It creates a barrier to make sure we have enough evidence before we implement change. Hopefully that makes sense. But we'll move right along now. So a knowledge check. Here you go. A researcher wants to test whether a new focused playlist helps students perform better on a 20-minute reading comprehension task. Students are randomly assigned to one of two groups. Group A reads in silence. Group A reads with the playlist. Which of the following statements represents the null hypothesis for this experiment? So again, null hypothesis. So we'll look at this as no change, or there's no, we'll say no change. We'll say no effect. So going into that is number one, the focus playlist will improve reading comprehension scores. That seems to be an effect. Number two, there is no significant difference in reading comprehension scores between students who use the focus playlist and those who read in silence. That sounds right to me. So students reading in silence will perform better. That shows me there's an effect. So I'm going to say that's a no. We'll cross that one out. Cross one out. The focus playlist will distract students and lower their scores. That shows me there's an effect. So lower. We have the word better here. You can see that's an effect. Improve, that's an effect. Number two is the only one that shows no effect. So going into this, when we're looking at observation, again, this is the big thing you're looking at here is when you're talking about observation. When I'm observing something, this is what kind of follows after that. You have two types of reasoning here. You have inductive reasoning and you have deductive reasoning. The one that we kind of use more often than not, and that I think people are more familiar with, is deductive reasoning. And if you're a fan of any literature, this is what Sherlock Holmes uses. I'm a big Sherlock Holmes fan, so this always resonated with me. Um, so a way to look at this is, and we'll start with deductive reasoning, is you're using a general principle to predict a result. In other words, like let's just give an example of something of a general principle. Well, a general principle would be like gravity or something we know. Um, I'm taking a general principle like gravity, and I can use that to predict something specific happening. We'll use Isaac Newton and his apple as an example. I have gravity. If I drop the apple goes down. Deductive reasoning. I'm using a general principle to predict how this apple is going to happen. That's that direction. Inductive reasoning, and change my colors again, is the opposite. You have the apple, and you're inferring gravity. This is what Isaac Newton actually did right up here. Isaac Newton sitting under the tree, the apple fell, and he uses an observation to come up with a general conclusion in which he postulated that there was an interaction between the earth and the apple. And today we know that as gravity. That's inductive reasoning. So those two types of reasoning, being able to distinguish between those, I'll ask on your quiz. So keep that in mind. So here you go. A knowledge check. A scientist observes that certain plants survive in very salty soil. Over time, they conclude these plants have adapted specialized root structures. Later, they predict a newly discovered plant in the same habitat will have similar adaptations. Identify which parts of this scenario involve inductive reasoning and which involve deductive and justify your choices. So, in this particular case, a scientist is observing that, and let me, again, my yellow's not going to work for this one, so we'll go to red here. So, plants are surviving in very salty soil. Over time, they conclude that these plants have adapted a specialized root structure. So, right there, they're observing something, they're observing a specific event, they're going specific to something general. And anytime you do that, that's inductive reasoning. Specific to general. Boom, boom. Over time, they conclude these plants have adapted specialized root structures. So now that they have this general claim right here, let me change my colors again. Sorry, I'm going to do this a lot, guys. So now that they have this general claim, they're going to use that to predict. So they predict a newly discovered plant in the same habitat will have similar results. This part is deductive reasoning. Okay. So if I go from a specific event happening, come up with a general principle, it is inductive reasoning. If I use that general principle, that general claim, maybe like gravity or this root system, the general claim to predict what's going to happen with a new plant that's discovered, okay, that's deductive reasoning. Hopefully that makes sense. Again, questions, comments, concerns, send me an email. So this is something that I want you to just think about because I'm big on using the right words and the right understanding of words before we just absolutely go into knowing the words we use. Let me rephrase that. I want you to use the right words when you talk. And when I did my defense of my thesis, uh, my very first question I was told was the basic scientist should be able to distinguish between a hypothesis, a theory, and a law, and be able to use them properly. Now, again, I want you to think about this, just kind of stew on it. I don't necessarily ask this; it's a little early level, but just think about it. When you're looking at a hypothesis, you're looking at something that's a proposed testable explanation. When you're looking at a theory, it's a well-substantiated, comprehensive explanation. So many scientific tests have been done and that support this theory. A scientific law is something that is holds true under no matter what the conditions are. It states what happens, and it's often mathematically. So a good example of this is gravity is a law because we have a mathematical concept that explains gravity. A scientific theory would be something like germ theory. We know how germs work, and we have a lot of tests that have gone into that. There's nothing mathematical really about it, but a scientific theory, it holds because of the experiments that have been done to substantiate that. And a hypothesis is sort of this one-off testable explanation for any uh, for any experiment. Explanation. So remember that when we're looking at a theory, we're looking at something that, because again, the big thing with theory is in the layman's terms and in like everyday life, theory just means guess. But in science, theory does not equal a guess. It is something that's well substantiated. Germ theory is a big one. Scientific law, gravity, that's a big one too. Laws of motion, we can mathematically describe them. It explains how it happens, but does not necessarily explain why. So just remember that. I will not test you on that. I expect you to know what hypothesis is for sure. All this stuff here I don't really care about, but I just want you to have some food for thought. So then when we look at science, we look at two types of science. You have basic versus applied. Um, what we are doing in biology, it falls under the purview of basic science. Those of you who are going on to further studies, maybe if you're going on to research or you're going on to like maybe do like vaccine research or something like that, vaccine research or like pharmacy or something like that. Real world problems is a good way to sum it up. Real world problem is what you look at when you're looking at this kind of applied science right there. Basic science is kind of what we talk about. It's just expanding knowledge of the world we live in. So when we talk about the cell cycle, we talk about genetics. Um, those are things that when we talk about basic science, we're expanding knowledge. And that's kind of what you're doing today. Now, we can dovetail it a little into applied science. So we learn about genetics, but curing genetic disease falls under this purview. So you can find cures. That's the big thing. You can help with weight loss, dietary stuff, crop yield, stuff like that. Where basic is just we're just learning the nuts and bolts of everything, and that's what we're doing here today too. But I'll dovetail into some applied science too throughout the semester. Um, this is just a fun little story right here. Alexander Fleming, accidentally. So we have penicillin, and if you've ever gone to the doctor, and there's many types of antibiotics, that's ugly. Um, there's many types of antibiotics, but the first big one was penicillin. Um, basically, he ended up happening is he left some bacteria petri dishes open, and a mold grew on it that killed the bacteria accidentally. We call this the serendipity in science. This is probably one of the biggest examples of this is that there was this serendipitous event that happened, and we have and he didn't do anything for it. He just was there. So he basically revealed that there's a fungus that produced penicillin, and that's one of our most famous antibiotics. And there's other antibiotics now, but I just find this story really interesting because lots of luck can come or can be needed to get good advances here. Again, I don't need you to know Alexander Fleming's name. Just the serendipity and the blind luck is kind of fun to think about. Um, within the scientific community, we've talked about this, and there's a reason we have this. There's something that every scientific research goes through calling peer review process, and that's just when people look at your work. You send your research out to other people. So you see these lovely people here are doing research and they're peer reviewing. I have had the privilege of being a peer reviewer on a few journals where I get to review people's work and see what they needed to do. Once it gets past this part here, once it gets past peer review, it can move into publication. And once it's in publication, other people can start repeating it. So this is the community we work in here. You need to make sure you're clear in what you're saying. And this is kind of how I or sorry, I grade my writing. So whenever you're writing your lab write-ups, it should be very clear and I should know what you're talking about. So be clear. Uh, I would even be a little redundant in your writings. It's going to help me understand. I'm coming in from the knowledge that, or from the standpoint of I know nothing. So you tell me what you did. When we would do research or we would write papers, basically it's step one is tell them what you're going to tell them, then tell them, then tell them what you told them. Don't skip any steps. Be clear. If you skip steps, I'll point it out. And again, your writing's going to grow in this class, so don't get scared. Just trying to give you a heads up. That being said, there's also some scientific ethics issues that we see in that we've seen in history. In 1932, you had the Tuskegee syphilis study. That people, specifically African American men, were withheld treatment from syphilis just to see the effects of syphilis. Very unethical. You could not do that today. Henrietta Lacks, her cells, her cervical cells were taken without her consent. Um, Now these cells, so here's the thing. Her cells have been used for countless medical breakthroughs, but she was not informed. She had no informed consent. They were taken without her knowledge. So it begs the question, and again, this is how I think of things personally, and I think um, it's a good philosophical mental exercise, is the ethics behind it. And do the ends justify the means? So I'll just say ends justify means. We got a good result, but no consent. We learned a lot about syphilis, but people suffered, and no consent. Today, you would go through something called an IRB, Institutional Research Board, an ethics board, to make sure what you're doing when you design your experiment is ethical and it's not violating people's rights. Again, which is why these medical advances take so much time, because you need to make sure you're doing it right. And just because you get a good result does not justify the treatment of your subjects and your participants in your study. There's other ones you can look up: the Stanford prison experiment in 1971, the Guatemala STD experiments, um, the milligram obedience study, and then the hepatitis experiments. You don't need to know these. Look them up. These are real research. This is real research that people did, that violated rights on a number of levels. So there's a reason that we have the whatsoever. There's a reason we have the stops in place, and the reason we have the checks in place to make sure that these horrendous breaches of rights violations don't happen again. Um, So knowledge check. This is something that, again, you don't need to know. I won't ask you on this, but as you go into science, as you go into your medical fields, um, your patients, if you go into research, whatever you're looking at, people need to have fully informed consent to undertake any scientific experiment that they're going under or research that they're consenting to. They can't be high, they can't be on alcohol, they need to be fully informed. Um, you can't give away their identity. They have to be anonymized. And if you ever present a research that you're going to do to like an IRB, they're going to ask, how are you anonymizing the data? How are you de-identifying people? That's the independent ethical review, the IRB, the ethics committee. Any participant has the right to withdraw. And you're very transparent. And I think this is my favorite part of science is transparency. I should be able to tell you everything I've done without having the temptation to hide either results, methods, whatever. Transparency is key. Remember that. So we've talked about science. We've talked about this ad nauseum. Probably I have bored you to death with it. But what I promised we would talk about today is what makes something alive. Okay? Now, we can look at this, and part of your write-up will be to analyze the alive statement. You'll see it in your D2L in your D2L portfolio or your D2L account. Sorry, not portfolio. But what makes something alive? And these are the big things. This is kind of what the community kind of has a consensus of, that there is some form of order within the individual. So even if you go from bacteria to something like humans, there's order. Bacteria have a cell or a cell, not a cell wall. They have a cell membrane, and they have organelles, They have structure to them. Humans, we have organs. We have other stuff. We have atoms in us. There's an organization to us from the smallest to the largest thing. So there's order. That is one thing. But you can have order in a number of different things too. It doesn't mean it's alive, which is why, I mean, I'll stay on the red, is that anything that's alive usually has the ability to reproduce. Um, so they can create offspring with similar characteristics. Now, this does bring up a question. What if something is sterile? Does that immediately uh, does that immediately omit them from being alive, or does that take them out of the alive category? You could look at something when a donkey and a horse mate, they make a mule. And the thing about mules is they are sterile. Come on. There we go. Mules are sterile. So there's order, there's reproduction, a caveat being sterility, but they also grow and develop. And if they may not grow in size, but they may grow in number. Sorry, I'm losing my thumb here. So bacteria grow in number, cells. Come on. Sorry. CLLS. Humans get larger. They start off as a baby and then they become adults who take biology 1 at Lone Star. Energy processing. So in a number of ways, I mean, I'll start with humans. They use food. They ingest food, or some form of metabolism. They respond to stimuli. So a good one would be heat, cold, light. They respond to these things. Turn the heat up, you sweat. Turn the heat down, you start to shiver. Turn the lights on, your pupils dilate. Same concept. Adaptation. They can fit to their environment. Expose yourself to enough heat, your body will adapt and it won't sweat as much, and vice versa with the cold. Same thing with the light. Turn the lights off in the dark, and your pupils dilate, or they undilate. I don't know what the right term is. So you see better in the dark. Homeostasis means they just maintain stable conditions. Your internal body temperature is 98.6 degrees. That's an average. Some people go a little hot, some people go a little cold, but 98.6-degree average is how you maintain homeostasis with your environment. If you're unable to do this, you'll die. And then, as an organism, as a population, I should say, populations that evolve through mutation and selection is a good way to look at something as being alive. And again, questions on that, shoot me an email. And these are just different types of responses to stimuli. Plants, phototropism, they grow towards the light. When we talk about photosynthesis, you'll see why. But plants want the light. Sound. You hear a sound. Some animals, like bats, use echolocation to navigate. Touch. Venus flytraps, they'll shut in response to a fly landing in the mouth of the leaves. Temperature, I mentioned shiver and cold. Heat, they may sweat or they may move into a cooler environment. And then chemicals. Bacteria move towards certain chemical signals. Nutrients, toxins, fungi do this too. Um, and then even if you were to ingest the wrong chemical, your body responds. So fun stuff. Again, homeostasis in action. We don't need to go over this stuff. This is more of the same. Um, Going back to the levels of organization, that again, that's the one of the criteria that makes something alive is just they have that level of organization. Um, at the chemical level, we have atoms, molecules, nucleus, are just the building blocks of matter. At the cellular level, you have cells, you have organelles. Tissue level, you have your organs, or you have your tissue, you have your muscle or nervous tissue, I should say, and you have your organs like your liver, your heart, your lungs. Then you go to the organism and then the ecosystem level. This is how the systems are broken down. But that level of organization is paramount in uh, determining life. Almost there, guys. More levels of organization. Again, we can keep talking about that. Homeostasis in action. Polar bears, they're able to maintain their body temperature through that fur, and they have a lot of fat. Um, that's just one example. Again, I should be able to give you an example of an animal or an organism responding to its environment and maintaining an equilibrium of sorts. And you tell me, oh, that's homeostasis. Um, as we start going down bigger, I guess, as we start going down this biology tunnel into more specific areas of biology, you're going to look at life in general, and you have three classifications of life. Um, you have bacteria, you have archaea, and eukarya. Um, the two that we're going to talk about in this class are bacteria and eukarya. That is a terrible, terrible boot I just made. But essentially, what you're looking at is there are two types of life in the world, and we just look at these guys. Prokaryotes are the first one. Golly, I'm using prokaryotes and eukaryotes. Prokaryotes, no nucleus in their cells. Eukaryotes have a nucleus. This is where all the higher level organisms live. So we as humans are eukaryotes. If you don't have a nucleus, you're a prokaryote. Okay? So breaking down the tree of life, we'll go into bacteria, we'll go into eukarya, but we really won't touch on archaea. Um, this slide kind of filler. There's diversity across domains. You're going to see that whether it's plants, animals, fungi, bacteria, doesn't matter. You're going to those same principles of life are going to. There's diversity, but there's unity within that diversity. Fun fact, that's where university comes from. Just so you know that. So unity in diversity, university. Okay, which is where you are. Um, but yeah, so you're going to see the unity through life, the cat classification of the life, but there's diversity. A flower does not look like a lion does not look like bacteria, so on and so forth. And there's a few other branches of biology. I mean, if you want to go further down into this, I think most of you are probably going to go into the medical field, but molecular biology, microbiology, neurobiology, paleontology, botany, zoology. The beautiful thing about statistics is I get to go into any one of these fields and help researchers out. So there are certainly careers out there beyond the medical if you're interested. And again, biology, past, present, future. We're laying the groundwork for your biological knowledge right now. And as we go further into um, learning more about what makes us us, what makes the world, so on and so forth, you're going to learn new things. You're going to have questions. You can always ask them. Um, you're going to learn why we do things a certain way. Hopefully, you've learned a little bit in this lecture that we do things a certain way because it protects people. We as a civilization have made some bad mistakes, so would that have irreparable damage? Even though the results gave us good information, it hurt people. We're trying to avoid that hurt. So hopefully you're understanding some of this, and as we continue to go through um, the structure and the basics of biology, and as we grow and we go further and further into it, you'll understand the historical foundation, where this is leading us, and currently why we do things the way we do them. Like when we get into DNA, why we take certain drugs to attack cancer, and why that has an effect on us. You're going to learn that. So I'm hoping at this point your brain is firing and that you're very, very excited about what's to come. That's all I have for right now. So I'm going to let you guys go, and you have a few assignments to take care of and a quiz, but that'll be on D2L. I will talk to you later, and see you guys next time.